Today we'll be reviewing unit diabetes for the PLTW PBS midterm. First thing that we're going to look at is want to make sure that you know each of these terms. First up, we have insulin. Remember, insulin is a hormone, and it is going to be responsible for lowering blood sugar. When I see L, that helps me remember that we're talking about lowering. Also remember, a hormone is a protein. And remember, when we're talking about blood sugar, we're also talking about the glucose level. So insulin lowers blood sugar. This is also going to be made by the pancreas, which is an organ that is going to secrete insulin into your bloodstream. Next up, we have glucagon. Glucagon is also a hormone. This one works opposite of insulin. Therefore, it's going to be responsible for raising blood glucose levels. And when I think about this, I think glucagon has that same gluc, so it's going to increase or raise the glucose level. This is all produced by the pancreas. So for pancreas, we have a gland that produces, secretes insulin and glucagon. Next up, we have the difference between these two types of diabetes. First up, we have type 1 diabetes. Type 1 diabetes, you're going to have to be able to diagnose someone. Remember, blood glucose values are going to be greater than or equal to 200 milligrams per day. You might be given a graph, or you might be given a chart or a data table, and from that you'll have to do your diagnosis. The other thing to note is they make little to no insulin. Again, you'll have to be able to analyze graph, data table, or chart for that. Type 2 diabetes is when your blood glucose level, once again, is greater than or equal to 200 milligrams per day. In this case, though, you make insulin. But the insulin that you make is ineffective. Meaning it does not do a good job of lowering your blood glucose levels. Then we have the two types of feedback mechanisms. Remember, a feedback mechanism in and of itself is going to allow you to maintain homeostasis. Homeostasis is talking about balance in your body. One classic example is going to be body temperature, whereas if you're too hot, you're going to sweat. If you're too cold, you're going to shiver. A positive feedback mechanism is going to be when an action reinforces previous action. That'd be a positive to a positive, negative to a negative. The two of them are in a direct relationship. One example that we looked at for this was where when someone's in labor, they wind up having contractions. Why do they have contractions? It's because their body produces a hormone called oxytocin, which is a positive, and that forces the contraction to occur, which puts pressure on the cervix, which is going to be where the baby is going to be born from, and that causes more of the hormone, which causes more pressure, more hormone. So positive reinforces a positive reinforces a positive. Next up, we have negative feedback. Negative feedback is when you have the opposite action. So this would be a positive followed by a negative. The example that we use for this one is those blood glucose levels. When we're talking about blood glucose, insulin, glucagon, all working together. If we were to look at this, it'd be cyclical, up and down, up and down. Over a long period of time, it would balance out and average out, so that person is maintaining homeostasis. Reinforcing that feedback mechanism idea on the next page, it says feedback mechanisms have evolved to maintain homeostasis, as we just said on the previous page. what we have here is it says, describe how homeostasis is attained through feedback. In your answer, be sure to identify one feedback mechanism. So the feedback mechanism we spent a long time on was blood glucose. And other than death, what can happen? If someone is unable to maintain their blood glucose levels, they would wind up having diabetes. They also, short term, right, if they can't maintain their diabetes, they can deal with hyperglycemia or hypoglycemia. These terms we're going to review as we move through this packet. The next section that we covered was calories and food science. For food science, you have to be able to identify the definition for calories. Calories is the amount of energy in food. So when you eat a food and you look on that nutrition label, you'll see that there's something called calories. Those are really kilocalories. We're going to review how you actually go about figuring out chemistry calories and kilocalories as well. Over here we have the 4 major macromolecules that we have focused on. The macromolecules are large molecules. We also have something called a monomer. Remember, the monomer is referring to the building block. And then finally we have a picture of each structure. Definitely make sure you're able to identify both the monomer and then also the structure. First up, we have proteins. Proteins, the building blocks of those are going to be amino acids. When I'm looking at the structure of a protein, one thing that I always think is I look for that nitrogen. Protein has an N in it. It also has nitrogen. Lipid, remember lipid is one of those ones that's a gray area. It technically is not made up of monomers, but of a lipid are going to be glycerol and fatty acids. One thing to note about a lipid molecule is that there are many chemical bonds. Since there are many chemical bonds, it is going to have a high amount of energy. Remember, that's where that energy is stored, inside of those chemical bonds. Next up, we have carbohydrates. These are the fancy names for them: monosaccharide, disaccharide, polysaccharide. Remember, mono means one, di means two, poly means many. The building blocks of a carbohydrate can go by a few different. One is going to be a monosaccharide. Remember, that just means one sugar molecule. The other one you might glucose. And then finally, you might see simple sugar. When you're looking to identify these, note they have that ring structure. This one here would be a because it's made up of more than 2 sugar molecules. Note here it's attached by this oxygen. In order to make it, what had to happen? Synthesis or hydrolysis. They had to remove water. So just to differentiate between those two, dehydration, synthesis, remove water to build. Right? It's going to be something that we're making larger. That's how we make our polymers or macromolecules. Hydrolysis is going to be when you move the water in order to break down rest molecules. Finally, we have nucleic acid, DNA or RNA, which is what we're covering right now. The building blocks for that is going to be a nucleotide. Over here we have a chain of nucleotides. Note this is DNA. DNA is a double helix, 2 sides, twisted ladder. The nucleotides are going to have different bases, A, T, C, and G, or U if we're talking about RNA. In this molecule right here, right, that section of the molecule, that is one. Now, what happens when you actually have diabetes? What types of dealing with? There's the hyper and hypoglycemia. Here I have a picture. It says the diagram below represents a cell in the body of a person with diabetes. All of those little dots are representing glucose molecules. We have to be able to differentiate between the high glucose and the low glucose. Looking on the outside, you'll notice that there's a very, very high sugar concentration. High sugar means hyperglycemia. Remember, someone that's hyper would have, so there's a lot of sugar. Also remember, glucose isn't going to be going through that membrane in and out. The water is going to be. So because of that, whenever you have a high sugar concentration, relatively speaking, that means you have a low water concentration. Looking at question 1, it says, identify if the cell hyper, hypo, or isotonic environment. Well, like I said before, it's going to be hyper. And the reason for that is because there's high sugar outside the cell. The next question, what is it called when it's the diffusion of water? Remember I said H2, osmosis. So osmosis is the name, but I H2O, osmosis. Then it says, describe how the water would diffuse. When we're talking about diffusion, we're talking about things moving, remember, downhill. So downhill is going from a high concentration to a low concentration. That means that it is necessary for this to happen. Also remember, we're talking about the water, not the sugar. That's the number one problem that people have when they're evaluating this. We did say initially, low water on the outside. That means, relatively speaking, there's going to be high water in the inside. Diffusion is talking about moving high to low. Because of that, this water is going to move out. It's trying to reach dynamic equilibrium, homeostasis. It wants to be equal on both sides. So water would diffuse out. And this would cause the cell to shrink or shrivel. Think about the fact that now this person is going to be, their cells are all dehydrated. So if you have hyperglycemia, you're going to be thirsty all the time. All the cells, meaning that there actually is a really, really high concentration of water in your blood. So your kidneys are working overtime. This person's also going to have very frequent urination. The other thing is, all the sugar is on the outside. None of that sugar is getting in because the insulin is not properly. And because of that, they are going to be, these people are going to be very, very tired, and they're going to be really hungry. Next section. A woman was meeting her friend for lunch. Before she left her house, she took her insulin. Okay, so first important thing to note. When she got to the restaurant, there was a long wait for a table. At the time, the woman began to feel dizzy and confused. Her friend noticed she was pale and sweating and called for assistance. Was she experiencing hypoglycemia or hyperglycemia? What treatment would you recommend for her? So the thing to note is that she took insulin. That means any glucose that was in her blood was being taken up by those cells. Therefore, she had low blood sugar. She had something called hypoglycemia. Remember, when I think hypo, I think low. So hypoglycemia, she had low blood sugar. Again, all of that sugar was moving into her cells because she took insulin ahead of time and she hadn't ate any for an extended period of time. What should she do? She should eat something. She could drink something like orange juice that has a high sugar concentration. She could take a glucose tablet. She wants to make sure that she ingests something that's going to increase her blood sugar level. When we're talking about long-term problems that you might have if you cannot maintain your blood sugar. So frequently you're dealing with hypoglycemia or hyperglycemia. What can happen? First, kidney failure. They're overworked from filtering the glucose. Next, this is because poor circulation. When you have high blood sugar, your blood vessels are not going to be able to withstand that. That means decrease in terms of oxygen getting to those cells. Less oxygen getting to the cells, glucose is not getting into the cells, those cells are going to start dying. Another thing we have is neuropathy. Neuropathy is loss of feeling, and that has to do with the fact that you have nerve damage. Again, that all stems from the fact that you have poor circulation. We have ketoacidosis, or DKA, right? That's diabetic ketoacidosis. That's what the DKA stands for. So you have high ketones, and this leads your blood to be acidic. Why is it acidic? It's acidic because your body is currently not using gluten source of food. Instead, your body is using lipids, fats. And long term, that is not good if you wind up in ketoacidosis. And the last one we have is PAD, P-A-D, peripheral artery disease. And this is up of fat in arteries, specifically your legs. So this causes a lot of pain in your lower body. The next page is focusing on analyzing those graphs. You need to be able to do this. If you have, make sure you look at the old do nows, you look at the old tests, the old quizzes. It will be very similar to those questions that we've already seen. I've already done the graphs here. You really could just base this off of the data table as well. But if I look at patient A, what I'm going to say is that they are capable of making insulin. They are capable of making insulin. It's the green line. And then over here, their glucose level, though, goes above 200, right? All the way up to 210. This person is going to have type 2 diabetes. And you want to be able to take information from both the glucose and the insulin graph. In this case, their glucose level's over 200, and their insulin, they do make it, but their insulin is ineffective. It does not lower their blood glucose level. Patient B, that's going to be the blue one. What you're going to notice is that they also have a spike. It's above 200 once again. Insulin levels, they are relatively low. They don't produce really anything, and there's no spike once they are introduced to glucose. Because of that, this person's going to have type 1 diabetes. Again, be able to cite both values. They make insulin, they have high levels of glucose over 200, and in terms of insulin, they make little to no insulin. Remember, sometimes they will not be right at 0. The main idea is that there's no increase in insulin even when exposed to glucose. And that final person, C, what you're going to see is that their blood glucose level stays below 140. Remember, 140 is that number for prediabetes. Here, this is less than. So this person is normal. They make effective insulin in response to glucose. And the last page that we're going to work on goes right back to the food science section. This is when we did all of those indicator testing. You need to know what color the indicators are going to turn when they are exposed to certain macromolecules for either a positive test or a negative test. Here we are. What you're going to see gives you the colors that it turns. My advice, make sure you know what they're each testing for. Benedict's is for glucose. Paper bag is for a lipid. Iodine is for starch. Biuret is for protein. Going down the list, Benedict's, this light blue color, that's a negative. If it was positive, it'd be orange. Iodine, this is positive, blue-black. Paper bag, transparent, translucent. That's going to be a positive. Over here, purple. Remember, purple means protein. This food has starch, lipid, and protein present. And our last question deals with trying to figure out how many calories are in each one of these food samples. Remember, all we're doing is using this data. We have mass of the water times change in the temperature. Do we have specific heat of water? Give that. This is all about plugging in the values and then typing them into the calculator. Remember what I said after the last test? Make sure you are putting it into the calculator. It is very easy to make a silly math mistake and then wind up